Hello guys, welcome to tvashta. Today we have a question from CSIR NET December 2019 paper from section C, and it says the following observations are made on a 30 residue polypeptide. A. Unordered structure is observed in water, but a helical conformation is observed in medium of low dielectric constant. B. The peptide is resistant to degradation by proteases. C. Red blood cells are lysed by the peptide. And D. Beta-mercaptoethanol has no effect on peptide structure. And they have asked which of the following statements can be correctly attributed to the above observations. So the options are 1. The peptide is entirely composed of D-amino acids and is amphipathic. Second, the peptide is entirely composed of L-amino acids and is not amphipathic. Third, the peptide is rich in disulphide bonds between D-cysteines. And fourth, the peptide is entirely composed of L-aromatic amino acids. Let's discuss each of these observations one by one. So the first one says unordered structure is observed in water, but a helical conformation is observed in medium of low dielectric constant. Unordered protein structures lack a fixed three-dimensional structure. They are flexible and they can adapt different structures depending upon their medium. Generally, peptides with higher proportion of charged and polar amino acids acquire this unordered structure because of their interactions with water. Also, attractions between unlike charges and repulsions between like charges promote unordered structure. Then the statement is saying that helical conformation is observed in medium of low dielectric constant. Dielectric constant in simplest terms is a measure of polarity of any medium. For example, water has high dielectric constant, so it is very polar. On the other hand, a medium with low dielectric constant will be less polar or more hydrophobic. Now, not all peptides can form an alpha helix. The sequence of amino acids play a very important role in the stability of alpha helices. If similarly charged amino acid residue such as glutamate is repeated several times in the peptide, it will disrupt the alpha helix because of repulsion due to negative charge. So generally, hydrophobic and hydrophilic amino acid residues are periodically spaced to ensure critical interactions between amino acid side chains. For example, a positively charged amino acid present 3 or sometimes 4 residues away from a negatively charged amino acid will allow the formation of an ion pair. 3 or 4 residues away because there are 3.6 residues in one turn. And hydrophobic amino acids are also similarly placed to allow hydrophobic interactions. Another point is that side chains protrude from the helical structure, as you can see in the picture. So in order to stabilize the helical structure of a peptide in a medium of low dielectric constant, that is in a hydrophobic medium, the peptide needs to have non-polar hydrophobic amino acid residues as well. So what we can conclude from this observation is that the peptide is amphipathic. That is, it has both hydrophilic and hydrophobic amino acid sequences. The hydrophilic part is responsible for the unordered structure observed in water, and the hydrophobic part is responsible for the helical conformation in medium of low dielectric constant. Next observation is the peptide is resistant to degradation by proteases. This can only mean one thing: D-amino acids. Peptide bonds formed by D-amino acids are more resistant to proteases. In higher organisms, all the proteins are made up of L-amino acids. And I hope you're aware that the stereoisomers L and D amino acids are enantiomers. That is, they are non-superimposable mirror images of each other. Please note that here L and D does not refer to levo and dextrorotatory. I repeat, here L and D does not refer to levo and dextrorotatory. These enantiomers may look similar, but to a biological system, L and D amino acids are as different as the left and the right hand. As I said earlier, in higher organisms, almost all the proteins are made up of L amino acids. So, the enzymes are also specific for L amino acids only. And therefore, most of the naturally occurring proteases cannot hydrolyze peptide bonds formed by D amino acids. The next observation is red blood cells are lysed by the peptide. Probably this peptide can disrupt the plasma membrane, and there are various mechanisms by which RBCs can be lysed. So we cannot infer anything specific from this observation. The next and the last observation is ß-mercaptoethanol has no effect on peptide structure. SS-mercaptoethanol is a reducing agent, and it is widely used to cleave the disulphide bonds that form between the thiol or the SH groups of cysteine residues. So if ß-mercaptoethanol has no effect on the peptide, it means that there are no sulfide bonds in the peptide. With all this information, let's try to solve this question. The fourth option says that the peptide is entirely composed of L-aromatic amino acids, which is kind of incorrect because we discussed that the peptide is composed of D-amino acids because it is resistant to degradation by proteases. The third option says the peptide is rich in disulphide bonds between D-cysteines. Again, it is incorrect because we saw that ß-mercaptoethanol has no effect on peptide structure. The second option is the peptide is entirely composed of L-amino acids. Again incorrect, and is not amphipathic. Again incorrect, we saw that it is amphipathic. So that leaves us with the first option, which is the correct option. The peptide is entirely composed of D-amino acids because it is resistant to degradation by proteases, and it is amphipathic as we saw because it has unordered structure in water and has helical conformation in medium of low dielectric constant. So our correct option is first. Thank you.